Thanks for the introduction. Yep, I'm the newest faculty at Nemours. I'm also an affiliated assistant professor in the psychological and brain sciences at UD. And today I am going to talk a little bit about like what my lab does and where we hope to go. And with that, I guess I'll just start. So, just some brief background. Epilepsy is what I study, pediatric epilepsy in particular. It's the most common neurological disorder. About 1 in 26 people will develop epilepsy in their lifetime, and there are about 50 to 65 million people worldwide living with epilepsy. Interestingly and importantly for what I do, the incidence of epilepsy varies by age. So you can see here that the early life period is associated with a particularly high incidence of epilepsy. So in this population in particular, in this pediatric population, it's more than just a disorder of seizures. So a lot of these kids are going to be diagnosed with some sort of comorbid psychiatric disorder. So these are things like ADHD, autism, anxiety disorders, executive dysfunction. And when you test them cognitively, even if they aren't diagnosed with a comorbid psychiatric disorder, you find that they often have learning and memory problems and, problems in school. And importantly, these are a significant predictor of quality of life, even more so than the seizures. So in this study, roughly 80% of a population of kids with active epilepsy have at least one DSM diagnosis or a measurable cognitive impairment. So when we think of the causes of behavioral and cognitive impairments in developmental epilepsy, we need to understand that there's some sort of underlying etiology or developmental insult that leads to the epilepsy. And this causes abnormal neural network development, which leads to these seizures that we can measure on the EEG and we can see clinically, as well as interictal discharges, which are sort of subclinical, but you can see on an EEG, and cognitive impairment. So standard interventions usually target the seizures because that's what we can see and what we use to diagnose epilepsy. But I propose that if we can target this abnormal neural network development with some sort of novel intervention, that we would be able to treat both the seizures and the discharges as well as the cognitive impairment. So we use a preclinical model of recurrent early life seizures to look at this. And so basically what this model is rodents. It was initially designed for rats, and then I moved it to mice, are exposed to an inhaled convulsant, and we can kind of titrate the dose and the number of seizures and the time course during development that we give these seizures. And then we wait for the animals to grow up and we ask, what are the long-term cognitive side effects of these early life seizures? And we can test their behavior and their cognition in a number of ways. So they have spatial learning and memory impairments. They have impairments in attention. They have impairments in social behaviors as well. But the goal of all of this is to work towards a treatment for these impairments specifically. And so what I started off with initially was a drug called ACTH for a number of reasons, not the least of which being that in patients with epilepsy, with a particular type of severe epilepsy, it's one of the only drugs that's associated with a better cognitive outcome. So I give this drug during the time of the seizures. I treat the animals with either a control or the drug. And then I wait for them to grow up, and I use actually a fear extinction paradigm, so fear learning and extinction paradigm, so that I can record the dynamics of the neurons in the brain while they're doing this learning. So in order to begin to understand the mechanism behind the effectiveness of ACTH in preventing poor cognitive outcome, we initially started with this hypothesis that the ACTH was working not in, potentially in addition to this traditional pathway that involves corticosteroids. We hypothesized that there was an additional component of these receptors that bind ACTH in the brain. And the reason for this is that in a variety of other disease models, including ischemia, stroke, Alzheimer's, multiple sclerosis, agonizing these receptors in the brain directly, either with ACTH or with another drug, leads to increased neurogenesis, BDNF, increased growth factors, increased synaptic plasticity, as well as reduction of some of the pathology that is associated with these various disorders. And so is this in our model what's leading to improved behavioral outcome after these early life seizures when we treat with the ACTH? So to do this, to test this, we again just used a simple fear acquisition and fear extinction paradigm. And the reason for this is that we had shown in rats that the animals that had experienced early life seizures were significantly impaired in this task. And what we see here is that mice are also impaired. ACTH improves this impairment, but only when the receptor in the brain for the ACTH is present. So it loses its effectiveness in the knockout animals. Full disclosure, there's something really interesting going on with controls for this group in particular, but that's sort of outside of the scope of this talk right now. But that's sort of, yeah. So now that we have a treatment strategy that's effective in preventing cognitive impairment after the early life seizures, can we use this to understand the neural dynamics that underlie this improvement? So I've shown, or we've published before, that early life seizures are associated with impaired cognition in a number of domains, that this can be improved. Actually, I didn't show these data, but this is also published in 2 rat models and now a mouse model of early life seizures or hyperexcitability broadly, and that this requires specifically MC4R. So how? So the first thing we did was we looked at short-term plasticity in the prefrontal cortex in particular. And what I didn't tell you is that fear extinction learning requires PFC. And so we looked in this brain region to see what potentially was going on at the level of plasticity. And we chose to look at a particular type of short-term plasticity because it's been hypothesized in silico to underlie synaptic processing in this brain region. And so what we've shown, which is similar to what we had shown in rats, but this is now in mice, is that the early life seizure animals actually have an increase in this type of plasticity. But we show here for the first time that the ACTH actually completely resolves this. And I was a little bit shocked to see these data. They're relatively new. I collected them over the summer. But it was pretty exciting that the ACTH was having an effect at this level. So now we wanted to know what was happening in vivo while the learning was actually happening. And so to do this, we use electrophysiology. We use tetrode recordings to basically triangulate the signal of single neurons in the brain while the animal's freely moving so that we can build these, activity patterns of these neurons while the mouse is learning. So the way we conceptualize understanding neural dynamics in this context are basically as follows. So we think of temporal coding, so firing of the neuron in time with respect to firing of like a local field potential, so the sum total of all of the other neurons. We think about population coding, so how pairs or groups of neurons are firing together in time, and then rate coding. And this is most simply understood here in terms of looking at a neuron who's increasing its firing rate with respect to some stimulus. So you turn the stimulus on, it fires faster. You turn the stimulus off, the neuron fires slower. But the best studied example of this is actually in the place cell system in CA1 of the hippocampus. And so basically what this is these CA1 neurons that have an increase in their firing rate when the mouse or rat is in a specific spot in space. And what I hope you can appreciate from this GIF is that this all 3 of these types of coding are happening at the same time in the brain and underlie cognition. So it's not just about a single place cell, it's about populations of place cells firing over time while the animal is moving in space. So to quantify this, we actually used a GLM approach, and what this gives us is post-spike filters, which are kind of a surrogate measure of temporal coding, coupling filters, which are a surrogate measure of population coding, and event filters, which give us something that's akin to a peristimulus time histogram, and they measure rate coding with respect to task parameters. So we did this recently in a totally different model and basically showed that when you use these measures of coding in the brain to predict how an individual rat will do in one single session of a task, you get a pretty good prediction of the accuracy that that rat was going to have in that task. So we're taking the post-spike filter, the coupling filter, and the event filter here, and we're using that in a generalized estimating equation to predict whether or not the animal is going to do really well in that session or whether or not the animal is going to do really poorly in that session, and we get a pretty good correlation. And interestingly, the diseased animals shown here as the red dots and the healthy controls actually lie in the same distribution. So this suggests to us that, you know, this might actually be a targetable therapeutic approach, that we might be able to change these neural dynamics in a way that makes this sort of unhealthy brain look more like a healthy brain, and that might underlie, and that would be expected or be predicted to underlie better cognition. So we have a number of ongoing experiments in the lab now. Well, we did before we left, but we're starting up again soon. And basically what our questions are what do the early life seizures do to neural dynamics in the PFC during this poor extinction learning, and how is this altered as a result of ACTH administration? So switching gears a bit, we're also interested in astrocyte dynamics, and specifically neuron dynamics, and you know how neurons are talking to astrocytes in the brain in vivo. So the reason for this is basically that astrocytes modulate neuronal function. They are so important for maintaining ion homeostasis and blood-brain barrier integrity. They're important for setting up developing synapses, so neurodevelopment. They're important for structural and functional plasticity as a result of learning. They also directly control neuronal oscillations and synchrony, neurotransmitter uptake. So much of what the neurons are doing is controlled by the astrocytes. So our question is, how are the astrocytes affected by early life seizures? How are they altering the neural dynamics? And is this potentially also a druggable target at the level of the astrocyte? And part of the reason for this is that I had showed before, I kind of glossed over it, but the MC4R, the receptor that the ACTH is binding to prevent cognitive impairment in this model, is also available on astrocytes. It's on neurons and astrocytes. So we have a couple of projects looking at this in vitro, and we actually hope to move this in vivo with some mini scopes to look at basically the same approach that we're using for neural dynamics, but now to add the astrocyte piece on top of it. So bringing this all back, you know, when we think about the causes of cognitive impairment in developmental epilepsies in particular, we know that there's some etiology, and interestingly, these astrocytes can per se cause seizures. So dysfunction in the astrocyte, not only the neuron, but also the astrocyte, can cause seizures. So understanding how these cells interact together dynamically over time could potentially help us understand something about this abnormal neural network communication more broadly, which leads again to the seizures and the cognitive impairment, and then may help us identify, you know, this novel intervention that we're looking for. So with that, I'd like to thank, so we work pretty closely with the Mahoney lab. He's now, Matt Mahoney's now at Jack's Labs, and he has two very talented graduate students. Jeff is actually working on a piece of this project that's related to gene expression. Rod Scott's lab, he also just moved to Nemours. His previous graduate student, Willie Curry, was really helpful in collecting some of the data for the one brain disease model with the first approach that we used, the first time that we used the approach, the generalized linear modeling approach. The Stafford lab at UVM, the Weston lab, the Krementsov lab, which they're working with Jeff as well to do some of the gene expression work that I didn't show. And then my lab, I had some really talented undergraduates and 3 really talented graduate students, and my previous lab manager, and my current lab manager, and then my funding. And I went through that really quickly, so please ask questions.